Howdy, my name is Mikaela Sebastyn, and I am a second-year veterinary student at the Texas A&M College of Veterinary Medicine and Biomedical Sciences. Today, on behalf of the PEER program, I will guide you through a clinical application of our One Health cellular biology module. For more information about our modules, visit our website in the link below. Let's apply our knowledge about cellular biology in terms of the bovid. For those of you who don't know what that means, bovid is another word for cow. We will be using the cow in our clinical application because it demonstrates the relationship between different cells to form a functional being. We will apply concepts of cell theory and cell types by looking at Trichomonas fetus and rumen acidosis. The cell theory consists of three main objectives. All organisms are made of one or more cells. Cells are the basic building blocks of life, and all cells come from pre-existing cells. Trichomonas fetus is a protozoa, or a single-celled organism commonly known as trick. Its life cycle can be described as binary fission, a form of asexual reproduction. It duplicates its DNA and then divides into two new organisms. For those of you familiar with biology, it sounds similar to mitosis, which applies to cells rather than organisms. Trick resides in the prepuce of a bull and causes asymptomatic infections. This means a bull can present with no clinical signs. Transmission occurs from bull to cow during breeding. Cows who are affected may abort their calves and become infertile. This disease is reportable to federal agencies, and all bulls must be screened before breeding can happen. Cattle are ruminants, meaning they have 4 stomachs. The compartments of the ruminant gut include the rumen, reticulum, omasum, and apomasum. For our purposes, let's focus on the rumen, which is the largest stomach compartment. Its functions include storage and fermentation. This means its environment is suitable for the growth of various microbes. These microbes ferment within the rumen to create volatile fatty acids, which are an energy source for ruminants. The microbes within the rumen that create energy are both prokaryotic and eukaryotic. This means we can find bacteria and protozoa. Check out the protozoa moving in this sample of rumen fluid. This is normal, healthy fluid. All of these organisms are essential for the health and well-being of the ruminant. Now, what would unhealthy rumen fluid look like? Note the lack of moving protozoa in this sample. This is not normal and could be a result of rumen acidosis. The normal rumen pH is 5.5 to 7. If the pH drops below 5.5, then acidosis will occur. The cause for acidosis is usually an increase in the level of rapidly digestible carbohydrates, such as grains. This leads to 2 major events: decreased rumen contractions and changes in the microbiome of the rumen. This means the good bacteria and protozoa die off, while the bad bacteria grow. Here is the sequence or pathogenesis of rumen acidosis. A decrease in the pH leads to damage of the rumen lining, which leads to the death of rumen microbes. This can be followed by many clinical signs from the ruminant, including decreased feed intake, minimal rumination, diarrhea, lethargy, and death. Treatment of rumen acidosis involves diet changes and replenishing the lost microorganisms. Look at this picture in the top right. The device on this cow is called a fistula, which has been surgically placed to allow access to the rumen. Thanks to this device, the rumen fluid of this healthy cow can be transferred to a cow with rumen acidosis. Consequently, the microbes will reproduce and restore the cow's health. This concludes our video. Thanks for watching.